I want to go back to the beginning, talk about your growing up in New York City and a little bit about your education and how you got interested in science.
I probably got interested in science when I was taken to a well-known physician because I had allergies. And he allowed me to see my hand in his fluoroscope, something that wouldn't be allowed any longer, but I put my hand in, it goes in between the source of the x-rays and the screen, and there were my bones, and I thought, this is cool. Or we didn't have that expression, the equivalent. And, but of course, no one knew what it was to be a scientist. That wasn't exactly a profession that was well known among people growing up in New York City in the 1930s. And so I thought I wanted to be a physician. And my parents thought that was great. And this went on for a while. But beginning in my early teens, I became very interested in science. And I went to Stuyvesant High School, which was still is, the most famous now science specialty high school in the country. And there I met a whole bunch of other kids, boys, because it didn't have girls then, who had my own, also shared my peculiar interests. So that was the beginning. And then I was encouraged to enter the Westinghouse Science Talent Search. And I won a second prize, which meant I came to Washington. And amusingly, they took us, there were 40 of us, they took us all out here to the NIH campus. And I believe I was taken to Building 2. And we were taken to some labs on the top floor. And I said to myself, if this is what it means to be a scientist, I'm not going to do it. It was pretty grubby. And of course, as fate has it, that's where I ended up as a scientist when I came to NIH. So at least for part of my career here.
Well, hopefully it's not so grubby today.
Well, but it's not a lab anymore.
Right.
It's an administrative office.
So you must have made your decision for your undergraduate work, something pre-med.
Well, that's exactly right. But at the Westinghouse Science Talent Search, and I was just thinking about this the other day, there were various people who talked to the prize winners, and a couple of them homed in on me and said, you don't have to be a doctor, you can be a scientist. That is a career. And that was news to me, because no one in my family or in any of their acquaintances really knew. I remember at one point when I was studying chemistry, a friend of the family said, what does that mean? You'll run a drugstore when you're finished, right? That was it. That was not an uncommon response. Anyway, so I went from Stuyvesant to Harvard. And at Harvard, I found a tutor named John Edsall. John Edsall became the editor of the Journal of Biological Chemistry and was one of the earliest people to apply physical chemistry and its way of thinking to biological problems. So John took me on as tutor, and for 3 years I met with him once a week, essentially one-on-one. And we read through as many advanced books as I could read through in my spare time, because I also had courses, of course. And slowly he began to talk about himself, which is very unusual for a Harvard professor to talk about his personal life. Not done. But his father had been dean of Harvard Medical School, and his father had forced him to go to medical school, he told me. And he said, you don't have to do that. And you should go to graduate school. And my parents, of course, my father said, science is a hobby. It's not a full-time job, and you'll never make a living. So I applied to 2 schools, to Caltech for graduate school and to Harvard Medical School. And I was accepted to both. And I went to see a very famous professor of medicine there for an interview.
At Harvard?
At Harvard Medical School. And I told him my concerns because they were going to give me a scholarship. And he said, you know, if you really want to do science, I wanted to do science. And it turned out I couldn't because, you know, I'm a physician. But if you want to do science, don't come here. Go to graduate school. If you change your mind in the future, we'll take you. So I'm just wondering whether that offer is still... It might be nice to try an alternate life just to see where it would lead.
That was pretty early to go for a PhD. There wasn't a lot of that.
Oh, there wasn't a lot, and there's a good reason. So when I went to Caltech, my thesis advisor was Linus Pauling, a very famous chemist. He had just been president of the American Chemical Society and given a famous speech in which he said anyone who chooses a career in chemistry should take vows of poverty. And I thought, oh, that's wonderful. Yes, it sounded, you know, a life dedicated to poverty and knowledge. Well, fortunately, it didn't turn out that way because just about the time I moved there, the U.S. began to fund scientific research in a big way. The National Science Foundation, which was at that time a pretty small operation, started to ramp up. My first year, I was a teaching assistant, and that's how I made enough money to live. But by second year, the NSF had began its pre-doctoral fellowship program. I got one of those. And I think actually Caltech graduate students got a rather large proportion of them, but there weren't that many.
Is this the late 40s we're talking here?
No, here we're talking 1952.
Okay.
I graduated Harvard in 1951. So, so there I was, and I had been told by my tutor, John Edsall, that the future of biology was in application of physical methods. And Pauling was essentially a physicist, although of course he applied himself to biological problems. And I actually ended up working on a theory of ferromagnetism, the quantum mechanical theory, which has nothing to do with biology, but it's okay, it was discipline.
Was that assigned? Did he assign
Well, it was kind of complicated. What Pauling did was he generated ideas at the rate of perhaps 5 a day. And his secretary would type out the suggestions for research on little slips of yellow paper, I remember. And they would go in your mailbox, and not only in your mailbox, in the mailboxes of all the faculty. And many of the faculty became famous for actually pursuing the suggestions that appeared on these little yellow slips. And the advice I got from one of the senior postdocs was, just read them, discard the ones that don't seem sensible. When you see one you like, do it, and forget all the rest. And that's what I did. This one appealed to me, I don't know why. And the fact of the matter is, I can't understand my thesis now, my PhD thesis. It's full of mathematics, it's full of physics. And I was pretty good at it, and I actually thought for a while about making that my career, even though I was really interested in biology. And the driving force there was, there weren't that many jobs. So one of the questions was, you worked in the play, just as we do now in the marketplace. You choose a career that matches the job market. That was the way it was. But one day I went to see Richard Feynman, the physicist, whom I knew because he knew all the graduate students, and even though I was in chemistry. And I said, I can't make up my mind whether this is a good thing to do, to stay in quantum chemistry. And he said, well, can you foresee any simplifying assumptions that would allow you to solve the problems in a relatively straightforward way, which was, of course, one of Feynman's great contributions to physics was to solve really complicated problems by rather remarkable methods. And I said, no, I didn't. I thought that actually the way it was going was computers, because we were just beginning to see digital computers, and the kinds of problems we had in chemistry would best be solved that way. And he said, well, if you can't see a simplification, get out. And so I did. And that's not quite true, because actually, so what happened was then I finished in 3 years. I got my PhD, and Pauling said, I won't let you do a biology postdoc. I won't let you. In those days, your thesis advisor was essentially in loco parentis or more. And he said, you have to have another year of theory, so you have to go to one of these 3 places. And I ended up then going for a postdoctoral year to Oxford to work with a man named Coulson, who was well known for studies of molecular structure, theory of molecular structure. And actually there I worked on the structure of the chlorocuprate anion, which I was able to predict using the so-called crystal field theory. It's my last theoretical contribution. That one I do understand.
So is this straight structural chemistry at this
point?
Well,
not structural chemistry. It was the chlorocuprate, I might as well say, the chlorocuprate anion is a copper 2+ with 4 chloride ions around it. So it has a net charge, negative charge of 2. And you expect it in principle to be tetrahedral, but there were forces having to do with the splitting of the d orbitals of the copper, which causes the whole thing to flatten. But when it flattens, the chloride ions repel each other, and that forces it back the other way, and there's a compromise between the 2 forces. And I calculated that, and then the next year, then someone did the structure, and I was right to within ½ degree. I was very pleased with that. So that was my last. So then what happens is, well, before I go to Oxford, my draft board calls me in because I have to get permission to go. Like you weren't free to travel without, once you finish your education. And I remember this very well. He was a lovely gentleman, and he liked, obviously, very kind, and he said, yes, well, we're going to let you go to England for a year. He said, but there's something we would like you to do while you're there. Yes, sir, I said. He said, we'd like you to get a commission in the military, because otherwise you're going into the infantry when you come home. Well, I took that to heart. And so while I was in England, I began to make arrangements. And what actually happened was I had gotten to know Alex Rich, who was became a well-known scientist. He was a postdoc of Pauling's at the same time I was a graduate student. And Alex got a position, and Alex also was in the doctor's draft, so he had to have a commission. He had to he joined the public health service. And as the clinical center opened, it was just opening, brand new, building 10, he got a lab there, and he invited me when I came back. He invited me to join, along with another colleague from Caltech by the name of David Davies, who became an eminent structural biologist. So I returned from England, having solved the chlorocuprate anion problem, and immediately began working on problems with nucleic acids.
Is this NIMH officially?
This is now, we're the Mental Health Institute, that's right. And a lot of interesting things are going on in NIH at that time. Childhood leukemia, Emil Freireich and his colleagues are working, I think, a floor or two above or below us, I can't remember. But I do remember the elevators being full of awful sights of children and distraught parents. I think of that and the revolution that was happening to treatment of childhood leukemia right under our noses at that time. We didn't know how exciting that was.
How did you get to nucleic acid?
Well, mainly because Alex had struck, Alex being a very social type, had struck up an acquaintance with Severo Ochoa. Ochoa won the Nobel Prize, but at that time I don't think he had. No, he hadn't. Because in fact, what he won the Nobel Prize for was the discovery of this enzyme, as I recall, called polynucleotide phosphorylase. So the name is correct. It's an enzyme that degrades RNA, but who knew in those days? And he and his colleague Marianne Grunberg-Monago, who became an eminent French scientist, they drove it backwards. So they started with ADP or UDP, which is, and the enzyme, and drove it backwards. You ended up with polyadenilic acid or polyuridylic acid. And Alex began to study those. And it turned out that when you mixed poly A and poly U, you got a two-stranded structure. And Alex was doing X-ray diffraction of fibers, and he and David Davies drew fibers of poly A and poly U, and showed that they had a duplex structure, as I recall. It's not the same as DNA, but it's closely related. Anyway, my job was to study the physical chemistry. So I began to do experiments. I had actually had some experimental experience as an undergraduate because I did an honors thesis at Harvard in John Edsall's lab, and I had some lab experience. My job was to study the kinetics, how fast did these things react, and exactly what was the stoichiometry. It was 1-to-1, one strand of A and one strand of U. And so I did this, and I measured the kinetics, which were tremendously fast. And then one day, I looked at the data, and it didn't seem to fit 1-to-1. And I repeated the experiment, and it still didn't fit 1-to-1. It looked as though it might be 2 strands of U and 1 strand of A. And David Davies and I were in a small room together, and I said to David, is there any way there could be a 3rd strand accommodated in this structure? and there were no computers, but we had models, and these models were these brass rods bolted together and big brass plates representing the bases. And David said, well, let's go next door and look at the model. It's the sort of thing that it happens very seldom. And he had a poly A, poly U duplex. He picked up an extra U, and he said, let's see, does it fit here? No. Oh, look, it fits here. And that was it. That became then the first triple-stranded structure. And also the second, that U was in what became known as the Hochstein position, except it was quite a while before Hochstein identified it in another structure, but we didn't know what to call it. It was the third strand. And so then Alex and David and I published this short note, I think in Journal of the American Chemical Society, and that's another thing to remember. There were very few places you could publish any of this stuff. There were very few journals, first of all, and there were the term molecular biology was just being invented, and the Journal of Molecular Biology didn't exist yet. So you published either in the Journal of Biological Chemistry, which was a bit skeptical about whether this was really biochemistry, or you published in the Journal of the American Chemical Society. That was about it. On the other hand, the advantage was that you only had to spend 1/2 day a month reading literature. That was it. So,
did you coin the term FDR triplex?
No, that was a Russian colleague who called it FDR. That did not occur to us to call it that. We didn't call it by our names.
So what's the significance of that?
Well, that took a long time to figure out. It's been now a large number of years, and it's actually only in the last few that it's become clear that triplex structures have important biological functions. They function in the organization of certain kinds of RNAs, and actually a couple of years ago, or maybe last year, we published a paper in which we showed that in the control of human beta globin gene expression, there's actually a feedback mechanism in which the RNA that's made has a segment, the globin RNA, that forms a triplex with the DNA of the regulatory region of the globin locus. And so if you, the implication is that if you overproduce that message, it will feed back on itself and slow itself down, so it's autoregulatory. But there have been actually quite a large number of other papers now showing triplex formation as an important regulatory mechanism. But you have to be patient.
So what was the reception to this paper then if you came out with this triplex? But the question is, what does it mean?
Well, nobody worried too much about that. We were in the gee whiz stage of molecular biology, and it got a lot of attention. And then people said, we don't know what it means. And you have to remember that there were no ways to study function in living cells yet. All these wonderful methods we have now just didn't exist. And of course, until we had DNA sequencing, DNA and RNA sequencing, a lot of this was very foggy.
So shortly after that, you decided to go to Pittsburgh.
Yeah, I served my, whatever the number of years was, I guess, 2 or 3 in the public health service. I had fulfilled my military obligation, and it was time to go out in the world, and there weren't too many places to go. But Pittsburgh had a biophysics department, and that was unusual. And it seemed like it might be a place to start, so we moved there. I applied for and got an NSF grant and then an NIH grant, and I remember very well that I filed my NIH application, and the person who managed the grant, my grant, here at NIH phoned me up and said, "You know, you haven't applied for enough money." How quaint.
Yeah, what a nice privilege to...
Well, in those days, you know, they were looking to expand.
Yeah.
To expand, I mean, that wouldn't happen to anybody today, not even a Nobel laureate.
What was your area of study? What was your project?
Well, I could, now that's interesting. So, two things. One is I became very interested already when I was still here in the whole issue of what stabilized multi-stranded structures, what were the energetics, because I was really a physical chemist. And my goal was to apply physical methods to biological systems. So, I began to study the effect of ionic strength on stability, and showed that, you know, you had to raise the salt to get the two things to combine. You had to raise it more to get the third strand on, because you were packing a lot of negative charge from the backbones in together, and you had to screen it off with more salt in the solution. That was the kind of thing I began with. At the same time, I went back to my interest in metal ions, because it was early days for DNA. The structure of DNA was published in 1953, which is my second year of graduate school. Its impact was not, it took a while to be felt. And I thought metalloproteins are a way for me to apply my quantum mechanical background. And at one point here at NIH in the evening, I taught a course on quantum mechanics applied to biology. That's what I thought was the future. And so in Pittsburgh, I had a graduate student who studied hemocyanin, which is a copper respiratory protein found in crustaceans, horseshoe crabs, lobsters, squid, and we actually did chemical experiments that strongly supported the idea that you certainly don't have heme in hemocyanin. You had a pair of copper ions bound to the protein, spaced just far enough apart that the oxygen molecule could span the distance between the two coppers. That was the model we proposed, and years later that was confirmed by much more direct methods. But that was sort of nice. But what happened was, so I pushed that in parallel. I had one grant for that and one grant for studying the stability of nucleic acids. But not surprisingly, what happened was that the nucleic acid stuff became more and more interesting, and clearly was the thing that was going to be the future of biology, and so more and more time went into that. And so then, after 2 years at Pittsburgh, it became clear that the plan that had been presented to me, that the department would expand and a lot of young people would be brought in, was not actually going to be executed. I was it, and that wasn't going to work. And just at that point, my colleague Marty Geller, who's still down the hall, and a couple of other friends came to a meeting in Pittsburgh, and walking along after a session, Marty said, "Do you still like it here?" And I said, "No." And he said, well, we're forming a new lab at the NIH, and maybe you'd like to come back and join us. So in 1961, the Laboratory of Molecular Biology was founded. Hans Stetten was the scientific director, and he did something really remarkable. So there was a big chunk of space and quite a number of positions available, and the reflex thought was, you hire an eminent scientist at the top of his career and let him organize a big lab with his minions. And there was a young guy, a friend, named Gordon Tompkins, who was already maybe a section chief here, I'm not sure, but perhaps not even that, who persuaded Stetten that instead he should let Gordon organize a new lab with a lot of young independent people and just let them go. And Stetten agreed to that, and so I came back. David Davies joined, Martin Gellert, Bruce Ames, who was an eminent geneticist, still alive. Todd Miles, who's an organic chemist, who's now retired. And
was Tompkins lab chief?
Tompkins was the lab chief. And we invited Harvey Itano. So Harvey Itano had been at Caltech with Pauling, and Harvey was the discoverer of sickle cell hemoglobin. He was the one who actually did the experiment showing that hemoglobin S was a separate entity with a different molecular structure. And interestingly, the Cancer Institute, to which Harvey was attached even when he was in California, evinced no interest whatever because it had nothing to do with cancer. Anyway, but Hans Stetten, sensibly enough, invited him here, and so he joined us, and each of us had a relatively small amount of space, but we had 3 or 4 postdocs, something like that, and the wonderful thing about that was that the fields were so small that we all had read all the literature for all of them. So we had, I think Fridays, we had our lab meeting. In those days, every lab, every group came to these meetings, and we all understood perfectly well. We took turns describing our work, and we all understood each other's work, and we all had useful suggestions to make about what the other should do, because we all had a grip on everything.
Are these all the labs in NIAMD?
No, these are all the labs in Laboratory of Molecular Biology.
Okay.
All of these small groups that had been brought together. It was an exhilarating time, because people would just be able to make wonderful suggestions for your own work, and you could do the same for them. And then slowly, of course, things got more complicated. The literatures got much more complicated and specialized, and gradually it became more and more difficult to make an intelligent comment about someone else's highly specialized kind of work, but we still do.
Did it remain that way through the 60s?
Oh, yes.
Okay.
Yes, and even now, when we all get together and someone presents something, people, because we have so much experience, many of us, so many years of experience. So to come back then to the issue of what I started to do, so I continued to work for a while on stabilization of nucleic acid structures through modulating the electrostatic forces. And then I started to stabilize nucleic acids, I started to use larger and larger positively charged molecules. So I had started with sodium ion, then I went to magnesium, then I went to organic charged molecules called spermine and spermidine. And then I thought, why not work with peptides? So I started to work with polylysine, which is a positive, all the lysines are positively charged. That's a big molecule, and sort of beginning to be like the proteins that are found in the nucleus, which are very lysine and arginine rich, those being positively charged amino acids. And so I started to use those.
And you're still looking to stabilize the multi-stranded?
Yes, I'm now looking at two-stranded, at DNA mostly now, and beginning to study the interactions and whether there's a preferential interaction of one over the other. And we published a number of papers like that. And then one day, because at the ends of these papers I began to write, and this is relevant to the study of the histones, which are the positively charged proteins found in the nucleus, which are rich in lysines and arginines. And one day, as I wrote this at the bottom of a paper, I thought, this is ridiculous. Why not actually work with these proteins? And so we switched, and we began to work with the proteins. And from there, very quickly, we began to work with chromatin, which is the naturally occurring complex of DNA and proteins, including the positively charged packaging proteins called histones.
It looked like you were working on the histones for a little while before you went to the chromatins.
Yes, yes. Well, of course, we thought the individual histones, everyone thought they might be. But even then, I began to think I'm not really as interested in the structural aspects, the physical aspects. Really, I'd like to know how this works, and I'd like to know how the chromatin helps specify which genes are active and which genes are inactive in the nucleus. So that would be about the time that Howard Cedar came to the lab, and Richard Edsall.
Is this mid-70s, something like that?
That's probably about right.
You can look up the exact date.
Anyway, and they were electrifying. Edsall in particular was full of ideas, and Cedar as well. And Howie and I in particular, Cedar and I, began to study chromatin as a template for RNA polymerase, and to show that it could be transcribed, that there were, and I became interested in how much of the DNA was accessible to transcription factors and to RNA polymerase, and that was the general direction in which the lab began to go. I'm just thinking about Richard Edsall in particular, who was a remarkable force in the laboratory.
In what way?
Well, he was, I'll tell this story. We had a program for MDs who were trying to, the doctor's draft still existed. And so we had a program where we offered postdoctoral fellowships in our institute. And the candidates who were successful came and they went around from lab to lab interviewing. And one day I got a phone call from Chris Anfinson, who was a Nobel laureate, who was in our institute at that time. And Chris said, you know, we had this guy come to interview, he said, and we told him what we were doing, and you know what he said? He said, you're doing the wrong thing. This is what you should be doing. So he said, Chris said, he said, you know, we don't like wise asses over here, but I know you do. So I'm sending him over. A little while later, in comes Richard. He sits down, he says, so what are you doing? I tell him, he says, you know, you're doing the wrong thing. And I said, I think you're right. And that's how Richard came to work in the lab.
Well, what were you doing wrong, and what did he help you do right?
Well, I mean, he wanted, as I recall, he wanted us to work more directly on templates, real templates, rather than artificial systems. He was one of the forces that persuaded me. And then Howie and I were the ones who actually did those early experiments on transcribing chromatin templates, as I recall.
What's the difference between the template, the artificial system, and
Well, I mean, you know, you start with DNA and then you add histones, and that's one way to do it. That's the way a physical chemist does it. The other way is you take a nucleus and you open it up, or you don't. I mean, like that. So, and you deal with all the mess.
That's what a microbiologist would do, I guess.
Yeah, well, that's what biologists do. And that's the common way to do things nowadays. Synthetic systems have their limits. And in fact, that was one of the problems, for example, with the histones. I don't think we had that problem, but other people did. Namely, people study the individual histones, and many of them had structures that you could form, you could solve by X-ray diffraction. And they have absolutely no relationship. Nature doesn't know about those. I mean, it's fine, you can make them in the lab, but it's the wrong way to go. And that's the kind of error that can happen if you do the reductionist sort of thing. If you start, as we now commonly do, with a whole cell, then it's messier, but you may avoid those kinds of errors anyway. Anyway, then the next step was to find a really good experimental system. And fortunately, there was one. So one of the problems in trying to isolate chromatin is you try to isolate it from most kinds of cells. Most nuclei are full of proteolytic enzymes and nucleases, so both the protein and the DNA and so you have to work very fast. In the early days, I like many other people, had been working with calf thymus chromatin. So the way you work with calf thymus chromatin is you go up to Baltimore with a bucket full of dry ice, and you stand there while they slaughter the calf, and they take out the thymus, which is sweetbreads, very good, delicious. And you immediately smother it in dry ice. You race back to the lab here, and then you spend the rest of the day isolating the chromatin and doing your experiments, because next day it's all gone. And that's because the thymus is full of enzymes that destroy chromatin. But probably it was Hal Weintraub, who unfortunately died of brain tumor quite young. Hal Weintraub and Mark Grudin, who's still active out at Fred Hutchinson in Seattle, they started to work with chromatin from chicken erythrocytes. So chicken red blood cells, bird red blood cells in general, keep their nuclei. We don't. So in the last stages of erythrocyte maturation, it spits out the nucleus, but birds and reptiles don't. And so, and chicken blood is easy to get. So we began to work with chicken blood and chicken erythrocytes. It's very easy to get nuclei from red blood. In fact, it was a demonstration. When our kids were growing up, I used to bring them in on a Saturday, and we would make nuclei from chicken red blood cells. We would take it to show and tell.
But they were the only ones in class with that kind of show and tell.
I'm not sure, you know.
Bethesda?
Bethesda, Chevy Chase
Yeah.
High School, absolutely.
So you're able to do your experiments at a more leisurely pace, or
Yes,
work in a more deliberate way.
Well, you could keep the erythrocytes in the refrigerator. You could keep the nuclei in the refrigerator overnight, probably even longer. We didn't, but there's no need to. And the chromatin that you made was stable, and we began to study the regulation of beta globin gene expression. And now things begin to get less fuzzy, I would say. So we identified regulatory sites, enhancers. In the early days when an enhancer was not a very well-defined thing, we identified an enhancer that was very important for red cell, for globin gene expression. And I'm trying to remember how the chronology of this, because now it all, I mean, given that I see it as a whole picture, now it's a little hard to remember the order in which things happen. But
my impression was that you discovered the regulatory region when you were still working with the histones.
Yeah, well, there is an important thing. What we noticed was that there was a DNA sequence in what would have been the promoter, what we thought of as, or at least upstream of the beta globin locus. So you have several beta globin genes that are developmentally regulated in the egg, and then in the early embryo, and then in the mature chicken, you get a different globin gene expressed. But there was one transcription factor, or at least a motif, that seemed to be shared not only by the globin genes, but by other genes associated with the erythroid gene, the general pattern of expression. And then we decided to look for this protein. And ultimately we purified it. Someone named Todd Evans, who's professor at Cornell now, Cornell Medical School, and we gave it the name, originally they gave it the name ERYF1, erythroid factor 1. At the same time, Stu Workin at Harvard purified it from mouse, and he called it GF1. Someone joked that he would never stick with that name because those were my initials. Anyway, there was a meeting. There's something called the hemoglobin switching meeting, which was a big, every other year. We met, and there was a mini conference of the interested parties.
About how many? About how many?
For this mini, well, the switching meeting has about 100, but there were about 6 of us in this mini conference. They were the interested people, and what we agreed by that time, it was becoming clear that this was the first of a whole family of proteins, and that they regulated not only erythroid expression, but one was involved in cardiac development, and a lot of different things. And so it was decided to name it after the central motif, which is GATA. Those are the 4 bases. We call them the GATA proteins, and our protein was called GATA1. And GATA1 turns out to be essential for not just for globin gene expression, but for a whole erythroid gene family, and the other GATA proteins are equally important in many different functions. So,
did you develop any of the other ones in your lab?
Not in the least, no.
Okay.
Because my focus was still on the issue of regulation of the globin genes and the chromatin structure. That was changes that were associated with it. There are a lot of papers in there that I would have to look at the bibliography to remember. But what stands out next, so we began to characterize the histone modifications that became and we were one of the earliest labs to do a rather thorough, there was a science paper we published on the modification patterns during development. But at the same time, we became interested in sort of a puzzle, namely that was it. One of the next things was we began to look on either sides of the beta globin locus, and we discovered that there was a big chunk of heterochromatin upstream of the beta globin locus. And then beyond that, there was another active gene, erythroid-specific active gene. So the question was, it was known that heterochromatin liked to spread, and that would silence everything. So how come it didn't encroach on the globin locus? And so we began to think in terms of there being some kind of boundary. And when we looked carefully, we could see that just between this big chunk of heterochromatin and the globin genes, there was a segment of DNA that seemed to be binding multiple proteins. And so we began to look at those proteins, and we characterized them one by one. And one in particular seemed interesting because, as I recall, that one alone was sufficient in our assays. We set up an assay to test if you had an enhancer, which is an activating element here, and the reporter gene here. Typically, that would be enough to turn that gene on, but we would then put a binding site for a putative blocker here and see whether that prevented the enhancer from activating the promoter. And in fact, we found that this one DNA sequence from that big block of stuff between the condensed chromatin and the globin locus, one segment of DNA binding one protein was effective in that assay. So we decided to purify that protein. And that took a while. But eventually we did, and that protein was a known protein called CTCF. What was known about it was some kind of transcription factor, and it had all kinds of funny properties. But what we then showed was that it was necessary and sufficient to prevent an enhancer from activating a promoter. And then what happened was interesting. So there was and still is a great scientist by the name of Shirley Tillman. She was running a lab at Princeton at the time. She ultimately became president of Princeton, which I've always considered a great loss. But she did a great job at that as well. But she had been studying for years. She was one of the 2 or 3 labs in the world studying something called imprinting. So imprinting is a phenomenon in which there are, of course, 2 copies of genes on the pairs of chromosomes, one on each of the chromosome pairs, and an imprinted gene is one which is only expressed from one or the other of the 2 copies and is transmitted either through the female or the male. So in the case of this locus that she was studying, called the IGF-2H19 locus, IGF-2 is insulin-like growth factor 2, and it's quite close to the insulin gene. In that case, IGF-2 is off on the chromosome copy that's comes from the female, and it's on the paternal chromosome. And the question was, how did that happen? And Shirley came to me at some point when she knew we were working on this enhancer blocking phenomenon, which is called insulation. Okay, we were working on insulation. She said, "I wonder." This was before we had actually discovered CTCF, but she said, "I'll bet there's an insulator, and that's how it's working." So that really focused our attention on that area, and I think we had just discovered CTCF, identified it, and she published the sequence of this region called the imprinted control region, which was the regulatory region which she had shown was determining and deciding which allele was expressed. And we took that. I remember I took the sequence, and I looked for a CTCF binding site, and I found several. And so we immediately began to look and confirm that, first of all, that CTCF bound, and it bounds to the maternal allele. And that would mean that the maternal allele would be shut down because the enhancer was here on one side, the gene was on the other, and the CTCF sat between them. But what Shirley had shown was that on the paternal allele, the DNA is methylated at that site in the imprinted control region. All the DNA is methylated. The methyl is at a C next to a G. That's the classic place where DNA gets methylated. Not the only, but the predominant place. And indeed, what we showed was if you methylated the DNA, CTCF no longer bound, so you no longer had an insulator. So then the enhancer could act. It's too simple by half, and of course nature leaves well enough alone. So it's a bit more complex than that, but that's essentially the correct story. Anyway, we then shared this information with Shirley because we would never have done this without her. And then they did it in the mouse, and we did it in the human, and we published it together in Nature. And that was very satisfying.
So CTCF was precisely the thing she was looking for.
Well, it was. She didn't know.
Yeah.
Yes, that's exactly right. She guessed that there was an insulator there, but very little was known. There was no eukaryote. There was no vertebrate insulator known. There was one in flies called suppressor of hairy wing, and that was the only, and maybe one other in Drosophila. But no one had ever found such an insulating element. Okay, so I mean that's a while ago now, early 2000s. But CTCF has become a sort of a growth industry, I would guess, because it's turned out that it's not only doing that simple-minded thing, but it's also helping to organize and we didn't do this, the large-scale organization of the genome. So it's been shown that the genome in eukaryotes is organized into big loops, and particularly in vertebrates, into big loops, and which are maybe a megabase in size of that order. And then these loops are closed off by a pair of CTCFs, and there are accessory factors that are actually causing the stabilization of the loop. So the CTCF is actually stopping the passage of cohesin. Cohesin is a complex that can link DNA strands together, and that's what it does. So the strands at the base are linked together. So you get big loop domains. And then regulatory elements in one loop domain find it hard to talk to genes in a separate domain, but they like to talk to ones that are in the same.
Did you do some of this looping investigation?
No, we did not. Not then, anyway. No, I mean, that's all work in a multitude of laboratories, and it's now a separate. We use the tools that have been developed a lot now, but no, that was essentially other groups.
Were you lab chief at this point?
LMB history. So Gordon Tompkins, we have to go back in time?
Yep.
That's all right. Gordon Tompkins was lab chief for about 10 years, it seems to me, something like that. He was charismatic and very funny, and had many, many job offers. In fact, at one point, he thought all of us would move to Yale to form the new biochemistry department there, and we all would have gone, except he decided against it at the end, so we all stayed. Then, after years of being romanced by all the major, being actually appointed professor at Harvard Medical School, as I recall, and turning that down, he finally took a job at UCSF. So San Francisco was expanding then. They had essentially had a fresh department, and Gordon was finally recruited there. And the fact is, Gordon was a terrible administrator, and we did all the administrative work. So when Gordon left, we asked that we be allowed to run it the way we had been used to running it, which was as a commune. And unusually, probably the only lab at NIH, we were allowed to not have, we had a rotating lab chief. Each of us served for a year. We met once a month, and we talked to each other. We always agreed with each other.
How many lab chiefs?
The section chiefs. These would be section
You're
chiefs.
right, section chiefs.
About 6, I would say.
Okay.
Marty Gillett, David Davies, Todd Miles, Bruce Ames, me. I'm missing somebody, but all right. And that went on for a number of years, very pleasantly. And then Alan Spiegel became scientific director. And Alan didn't, that was a good, he was probably right, because it was very intimidating, because when we wanted something, we showed up, all of us, in the scientific director's office, and that was difficult. So he said we had to pick somebody to represent the group, and that was me for 10 years or so. And it actually didn't change a thing, because we met once a month, we talked over what we wanted to do, and we agreed. We always seemed to agree. And then I just went and told them what we had agreed about. So, and meantime, the laboratory was, because I want to talk about the lab, it was a phenomenal place. Over the years, we began to accumulate members of the National Academy of Sciences. In those days, NIH had only a tiny handful. And we began to just get them, and not just staff members, but then slowly people who had been passed through the laboratory. I think we're up in the 20s now for present and past lab LMB alumni who were, or were when they were alive, National Academy members. And That's without actually trying.
What, you say it's without trying, but what do you think the cause was? Was there something?
Well, we
culture?
attracted tremendously smart, independent people, and it was a place people liked to work. But I think it was because we started out with, a tremendous group of people. I'm thinking of the original people: Harvey Atano, Bruce Ames, David Davies, Marty Gillett, and me. We were all Academy members. And that's usual, and not because we helped each other to get in. It was because we're in different fields. But it was just very, very high-powered science.
Do you think there was more opportunity because of the way the lab was initially organized?
Oh, absolutely. I think that it's this environment, and very supportive environment. In the outside world, so I was offered a professorship at Harvard Medical School, and I could see that there things are different. I mean, people didn't speak to one another. They didn't want their postdocs to speak to postdocs in other groups, and that's no fun. This is fun, and also it means that a postdoc who comes to your lab is going to benefit from being able to talk to all the other people in all the other labs. And NIH is altogether a culture of sharing ideas, equipment, techniques, which is not the way it is in most universities. Not at all. So that's why people who are here mostly stay. Bruce Ames didn't stay. Bruce moved to Berkeley and became professor at University of California, and he's retired now. He's emeritus, but I think he's still active.
David Davies stayed.
David Davies stayed. He died a couple of years ago. And Harvey Atano left. He went to San Diego, to La Jolla, become professor there in the medical school. He continued to study hemoglobinopathies of various kinds.
Any other LMB politics I should know about?
I can't really think of any. There weren't all that many. We were rather peaceful. There was no infighting. It's one of the wonderful things about NIH, because you're not really competing for resources in the way they are in the universities. People don't, they'll share with other people in a way that, and the other thing is that there's nothing to do all day long but talk science. You come in, you're not going to have a lot of committee meetings, no faculty meetings. We meet once a month for an hour to discuss lab issues. And then if we're recruiting someone, then of course we have a little bit more work to do, but it's trivial compared to the kinds of obligations you have out of the university system. So it's like that. It's fun to come to work every day.
But some of the structural things do make a difference, clearly. Let's go back to the science just a little bit.
Sure.
And talk about how you got involved in pancreatic science.
Ah, sure.
Because that's what you're doing now, right?
That's right, actually. Yes, we hadn't talked about that. We left when you asked me about long-range interactions and loop formation and so on. I did say that not until relatively recently have we been doing that. Well, someone came to the lab. I can't really remember how we. Of course, it's because of IGF-2. So, insulin-like growth factor 2 is not far from insulin itself. And there is a story associated with that. When we did the IGF-2 H19 locus and the printed control region stuff. I went to see Alan Spiegel to tell him this news, which was significant. And I said, I would like you to note that the word insulin is in there, and this is the Diabetes Institute. And Alan said, I don't want you ever to think that you have some obligation to work on something related to diabetes. You do basic research and you do whatever you please, but you don't have. I thought that was great, which of course only egged me on. So we looked a little, so as I said, IGF-2 is close to insulin. It's close to insulin, as these things go. And so we began, and people have begun to think about interactions between distant elements. We didn't have the so-called high C methods that dominate the technology nowadays. But someone came to the lab who was interested. His name was Zhisheng Xu, and just at that moment, the extramural part of our institute began to support islet preparations, so pancreatic islet preparation. So the reason for that was there was a big move to do islet transplants in patients. So patients with type 1 diabetes who had malfunctioning pancreatic islets, the thought was that you would give them transplants of, and that was quite popular, and the NIDDK intramural actually got a specialist to work on that. I think it's not so popular now because it turns out the immunosuppression problems are just as serious as they are for any other organ transplant. So the cure may have problems of its own. But in any case, they developed a whole series of centers that did organ preparation from deceased donors, and they began to produce islet preps, and you could sign up, and we did sign up, and that meant that we could get supplies of islets, not on demand, but whenever a donor appeared. And so we began to look at the insulin gene itself, and just at that moment, there had begun to appear methods for measuring contacts within the nucleus between a given site, and you could look at another specified site to see whether there was any kind of physical contact inside the nucleus. That was called 3C, and then that was upgraded to something called 4C, in which you could choose your anchor and then ask anywhere else in the genome, did that anchor make contact? And it was very early days. There was no computing expertise. For analyzing the data, we teamed up with Keiji Zhao's lab in the Heart Institute, and they wrote the software to do the analysis. And we began to take islet shipments and to measure contacts between insulin and other sites nearby sites on chromosome 11, which is where insulin is in humans. And what we discovered was that there were a lot of physical contacts, and then as an example, we chose one which seemed to be particularly significant. It was to a gene called synaptotagmin 8, SYT8. And what we showed was that the contact actually, when you fed the islets glucose, the contact strengthened. In this particular case, the contacts were stabilized by CTCF binding. And SYT8 turned out, no one knew what it did. We showed that it actually affected insulin secretion. So now we had a kind of complicated feedback mechanism in which the level of insulin expression affected the expression of an auxiliary protein, which would help. So when insulin goes up, SYT8 goes up, and now you secrete insulin more effectively. So that was the kind of physical feedback system, and it was the beginning of what we then did. There weren't many examples at that point of that kind of physical contact as a regulatory thing.
What was the next step then?
Well, the next step actually was the technology improved tremendously, and the software improved tremendously. And the postdoc who's still here, named Singh John, Xingjin, he extended that, and that we switched. Islets are, first of all, you really do depend on availability, and every batch is different in terms of how good the cells are, how healthy. But a cell line became available, which a human insulin cell line, which made insulin a beta cell line. And so we studied that, and he was able to measure the contacts between insulin and all the other chromosomes in the genome, and there are lots. And what turned out, which was amazing, is that a large fraction of those contacts are to sites that contain type 1 or type 2 diabetes susceptibility loci. So this
susceptibility what?
A locus that's associated with, from genome-wide analysis, GWAS data. Actually, in between there, we had gone with the islets. We actually had found still another gene called ANO1, which we then were able to show, which nobody had ever paid attention to, which we were able to show was mildly connected with a glucose intolerance in mice that had knocked out the gene. So this becomes a way to connect the physical genome to the regulatory genome. And so the analysis in the cell line shows, also enabled us as an example to identify a gene previously unrecognized as having something to do, in this case, with insulin secretion. So I mean, it's a way to think about the problem, and we're not going to try to do a comprehensive study. It's just proof of principle, if you like, and it's not just a proof of principle about how insulin regulates its environment, but it's certainly the way a lot of genes are going to operate. Insulin, it's interesting though, but insulin and globin are examples of two genes, the globin system, two genes that dominate the cells in which they are expressed. So a beta cell, one of the major products is insulin, and so you wouldn't be surprised that a lot of its strategies are devoted to seeing that insulin is made in just the right amount. And the same is true for erythroid cells, for red blood cells. Namely, you would expect, that beta globin, first of all, has to be matched to alpha globin, because the two chains have to be made in equal amounts. but the cell is going to devote a lot of its mechanism to homeostasis, to some kind of maintenance of a stable, productive system. So they may be special in that way, but the whole idea of physical contact, and we're certainly not the only people who thought of this. Peter Fraser, some years earlier actually, had proposed so-called transcription factories in which lots of RNA polymerases are clustered together and lots of transcription factors that are going to be shared by multiple genes are there, and all those genes clustered together in centrally located to share in those factors, which is an efficient way to do things.
So are you still publishing this work in this line of inquiry?
Yeah. Dr. John has been looking at issues having to do with long-range interactions in the insulin locus that are imprinted. So IGF-2 is imprinted, that's a distance away from insulin, and there are lots of questions about imprinting on the other side of the insulin gene, and he's just finishing a study of that. We're just writing it up.
Is this your section that's doing this particular
This is my
project?
section.
Moving up to the lab, it was interesting that the idea you were told you don't have to work on diabetes.
Yes.
And the laboratory of molecular biology can work on what it wants to work on.
Yes.
My question is, where do you see that going? Given the past, and you were there from the beginning, where do you see the lab going in the future?
Well, I mean, I think science has changed. The kind of science we did, there's still a place for it, which is people perceiving problems that are really not solved, and messy problems, and sort of clearing the shrubbery away to see the real problem, and then solving it. But one has only to look at a journal like Cell to see that the emphasis now, first of all, is very, very clinical. And secondly, in part because of that, involves massive numbers of people. So not too many papers have 3 authors anymore. 10 is probably, I don't know what the average number is, but in journals like Cell and Nature, it's typically a small army. And that's rather amusing, because in the history of quantitative biology, when I was starting out, we had a big influx of physicists. They came in for a couple of reasons. One is because they were under the misapprehension that the laws of physics, there would be laws of biology that were just as simple. Actually, the laws of physics aren't that simple anymore either, but they thought it would be simpler. But mainly, they came because physics had become big science. And if you read the descriptions of how these large-scale studies were, where 300 people arrive at the end of the study to a big auditorium where they're told what they found, which they didn't know, and then they all go home. So they thought correctly that biology was something that would allow for individual expression. And that's less true now. I mean, I don't know what it's like to be a member of a 50-person group. I mean, I'm sure people get satisfaction from it, but it's a different personality, I would say. I mean, the group leader still has, if he can actually remember the names of all 50 people, must enjoy it, and I know people who do, and they're wonderful. The science is fantastic. I don't want to impugn the science. Science is wonderful. there's so much of it that nobody can keep it in mind. And I spend a lot of my time now doing science, which we do our own experiments, but we can support them with millions of other experiments that have already been done. So you can ask all kinds of questions about whether two transcription factors are bound next to each other, near each other. That's one of the things we're quite interested in, CTCF cofactors, actually. And all that's in there, but nobody knows about it because the data were deposited in experiments and studies that were addressing other issues, but buried in there are also the answers to thousands of unasked questions. So that's sort of fun, but it's a different, why shouldn't it be different? I always do this experiment of thinking back an equal number of years. So if you think back 50 years from founding of Laboratory of Molecular Biology, it's 1911. And you think of what we didn't know. We didn't know anything. We didn't know anything about protein structure. In fact, we didn't know anything about protein structure until Linus Pauling. And that was when I was a senior in college. Up until then, there were all kinds of amusing proposals for what a polypeptide looked like. None of them remotely right.
So the lab took off shortly after that revolution, really.
As did many.
Yeah.
Yes, no question that the discovery of the structure of DNA and the structure of proteins changed the way biology was done. No question.
Well, is there anything else that we should talk about that we haven't touched on today?
I think talking about the future of science, if I were starting over, I think I would do neurobiology, because there's still the chance there, at least if you have good hands, to do your own experiments. For me, I worked at the bench until fairly recently, and that was the greatest pleasure. To focus on an experiment and to shut out everything else is one of the great pleasures. And now, there are a few places where that's appropriate, in a way. Everything is essentially pre-done for you. When we did our experiments, we made our own buffers first, which would horrify anyone. If I went out into the lab and said, make a buffer, I think they probably would be puzzled. So, but it's always fun. And if it's not fun, you shouldn't do it.
Well, this has been a lot of fun for me, and I've learned a lot. Thank you very much.
You're welcome.